The broadcast is now starting. All attendees are in listen-only mode.
Hey everybody, this is Dan Stevens, and we're just about to start Explore Zen. We'll give everybody, we'll give others who are not yet here maybe another 30 seconds, because we're right on the hour as we speak. While we're doing that, let me introduce Chantal Blanchard and Nicole Kuntz, who are co-presenting.
Hi, everyone.
Hi, everyone.
Nicole and Chantal are driving the mouse. Driving the mouse? I guess there's still only one mouse. Are there mice? Maybe there's mice. I'm not sure.
We've been with one mouse right now. Let's not confuse the issue.
The mouse is driving the cursor. I know there's one cursor. There's multiple mice and one cursor. Okay. If we're not here by now, then we're late. So let's get on with it. Before we start with the image analysis or the Zen content, we wanted to very quickly go over two resources, one of which is brand new and we're very excited to discuss and show to people. Chantal, you want to?
I'm doing the other one, Dan. Yeah.
Oh, okay. Somebody, let me have it.
Do you want to put the page up?
Sure.
Oh, sorry. I guess I could have done that, right? So the service page? No, not that one. The service page. Thank you. So hi, everybody. We just wanted to make you aware of this online support page, which has some valuable information. You'll see here that we have a disinfection recommendation. It's a one-page document. We also have a more complete cleaning your microscope and disinfecting your microscope document here in our handouts if anybody wants to download it. If you have any issues, just let us know and we'll gladly send it to you. There's also a 1-800 number here, which will allow you, where you can call, and there are PhDs or somebody that's run an imaging facility on the other end of the phone. You should get a human being anytime between 10 and 6 p.m. and they are able to help either through TeamViewer or Remote Assist. They can actually help you work through some of the questions that you might have with the software or your instrument. So we just wanted to make you aware that this is available. We also have the clean microscope document, which is essentially a standard how to clean objectives, etc. document. So please don't hesitate to log on to this website if you have any questions or you need any help. If you can't get a hold of us and you need something quickly, this is always a good place to go. Nicole?
Great. Thanks, Chantal, for sharing that resource. The second resource that we'd like to share with you is our ZEN knowledge base. So those of you who are here are ZEN users, I would assume, or general microscopy image users. So if I can just get to the knowledge base here.
Sorry, Nicole, that was supposed to be the homepage. There you go. Good.
Yeah, okay, so this is brand new. We've taken time at ZEISS to bring together a whole bunch of videos, webinars, tutorials by instrument. So those of you are using specific instruments. For example, an LSM 800, 900, or 980. There are multiple videos here, how-tos, for example, light path setup, explaining different tabs here. So just easy ways for you to try and figure things out on your own. If you wanted to do that first before calling support, a really great resource that a lot of our technical specialists and team have put a lot of time into bringing together for you. Here you'll see different categories, so stuff on Zen, different webinars that we've run, if you'd like to go and rewatch some of those, how-to guides as well. So very helpful and all in one place. So feel free to browse this area, and I think it'll be really helpful for you getting to know Zen and just getting comfortable with it and not feeling like you're scratching your head for something, let's say, super simple, like exporting a movie. So yeah, it's just Google Zen knowledge base and it'll take you right there.
No longer any need to attend the Explore Zen webinars because it's all there. So hang up, hang up now. So we watch
Right.
our attendance numbers plummet on the side of the screen here as we go.
Please don't listen to Dan.
Okay, let's dive into it. So we have an agenda today that includes shading correction, colocalization, spectral unmixing, the Connect module, which we'll get into the functionality of, some Intellisys, machine learning, image analysis, and then we're going to do very briefly a quick tease on some scripting and some online image processing, which we'll cover more next time we meet. So let's fire off with shading correction. Chantal, I think you're going to do the shading correction for us, aren't you?
Yeah, I am.
Great. So maybe we'll begin this by saying that this should not be necessary, what we're doing here. Always when you're acquiring images, best to set up your microscope so that you don't have any shading. So we shouldn't see these kind of tiled effect that we have here. This is a tiled brain. I think we have close to 1,000 images to create this tiled brain. Clearly, we have a tiling effect. It is possible to do a shading compensation prior to acquisition. There's a bit of a process to that when you're doing it for fluorescence that we won't go into. But if for whatever reason you're either unable to go through that process, you don't have the resources, you don't know how to, or you've already acquired your data and you want to do this shading correction, it is possible to do it post-acquisition. So we're going to do a shading correction from this tiled image. And in Zen, that's done through the processing tab with shading correction from tile image. Or sorry,
So the processing tab's here, yeah.
Shading reference from tile image. So once we've selected shading reference from tile image and we're going to use the active image, one thing you might want to apply that's not a default here is to use the Gaussian filter option. So, and it looks like we have to set the input there as well, Chantal. As Chantal presses go on that, I'll just explain what's being done by this because it will take 30 seconds or so, I think. So the shading reference is determined by taking all of these tiled images individually and averaging them through. And the concept is that the biological variation should disappear and all that's left is this constant shading. So this is the shading, the averaged shading image. And the reason we apply the Gaussian filter is because it blurs the tiles initially before we do that averaging. So it further eliminates the sharp contrast that the biological features generate. Now that we have an average shading reference, we can do the actual shading correction because we have an image and we have the corrected reference. So we're under the processing tab, selecting shading correction now, and we're going to select the brain as the input, and we need a shading reference that we've just generated from this same brain image. There we go. And if we apply that, we will get a shading corrected tiled brain. Plugging away here at the bottom, you can see the progress bar, almost done, and just calculating the final image here. Looks like I overestimated. I thought it was almost 1,000. It's looks like it's maybe more like just over 400. So there's the final, much improved over what the initial was. What do you think? Do we toggle back and forth or do a comparative diagram? There you go. Let's do something comparative.
Oh, sorry, I keep doing that, don't I? Multi-image, and I want to...
On the right-hand side, Chantal, the thumbnail.
Ah, I can't see it. Hold on a second. It is.
There you go. Oh, you got two of the same. Pick another
Did that
brain.
work? Oh, I thought did that work? No? There you go.
There you go. So the corrected is on the left, the uncorrected is on the right. If we zoom in here to see the tiled bits, you can see that we've got that all corrected. Okay, really quick and easy. I should say, I want to say that that requires that what you're seeing is in fact shading. So if you have some other artifact, it's not going to correct it. It has to be a tile by tile shading. But it's worth a try if you have a data set that you want to see if you can improve because you've got some fluorescence shading on it. Very good. I think we got that. Nicole, should we do the colocalization?
Sure.
So we had a number of, what we're starting with is all requests that we had as a result of our last Explore ZEN. So we had somebody requested shading, and I believe it was fluorescence shading. I hope we covered that. If we didn't cover your question from that and you know who you are, then please speak up, mention again. And we did have a number of requests for colocalization. So let's close all these images, Nicole, and open the colocalization image. So we've got an image here that Nicole's going to bring up that's green and red. It's actually a stack. So as we move through the stack, most of what we see at depth in the stack is just green, but there is this surface stain for the red, and there is some degree of overlap. So we want to take a look at how do we quantify that. So the easy way to present this, of course, is one channel's green, one channel's red, and I have some orange in my image. And that's a pretty common way to present something, say on a poster. But it can be difficult if you have something where you're comparing sample A to sample B, drug treatment, or you've done a wild type versus mutant, something like that. If all you've done is disturb the degree of colocalization, then it's rather difficult to convince somebody that you have more or less orange. So in that case, you want to be able to quantify it, and you can do that through the colocalization tab that's on the left-hand side of the image, about 2/3 of the way down there. Yep, terrific. So we're in colocalization, and we can see this scatter plot now. Nicole's going to bring us back up to kind of the top of the stack where we have two different colors. Terrific. And let's also display the table right from the beginning here, Nicole, if you don't mind. So there is a statistics table that you find in the colocalization tools, just to the right of that button, Nicole, the little checkbox.
Oh, right. Yep, there it goes.
Perfect.
All right.
Okay, so let's talk about what we're seeing and how we're measuring colocalization. So if you imagine, not imagine, it's actually the case that all of these pixels have an intensity value both for green and for red. But if we do the imagining part and we kind of discard all the spatial data from our image and we just have an array of values where the green and the red are paired up, and we plot those on a scatter plot where the x-axis is one fluorophore and the y-axis is another, we get the plot that we have right here in the screen on the left-hand side. So we have on the x-axis the beta-catenin, and the y-axis the ZO1, and we've got a correlation between the two. So all we're seeing here is when I have intensity of beta-catenin that is 11,700, what do I have the intensity of ZO1? And I've plotted all the pixels on here. And the concept is that if we have colocalization, we should see some correlation between these two. So we're going to have some population of pixels that have both fluorophores in them, and two fluorophores in one pixel means that we have something that has intensity for both channels, and so we get a line that travels at kind of a 45-degree angle in this scatter plot. If we didn't have any colocalization or we had an exclusion of it, then we would see two distinct lines going along the axes, which means there's no coincident stain. We have a measurement of that though. And in order to take a look at the measurement, we have to talk about the thresholding of these values. So the current scatter plot is analyzing every single pixel in the image, which is not so useful because now we have statistics of our background. We don't want to be analyzing our background because the degree of coincidence is going to be weighted according to how much sample is in the image. That doesn't make any sense. So we have to set some threshold here. So we're only doing the analysis on pixels that are above the background level of intensity of the two channels. And that's where the threshold is, where Nicole's got her mouse over right now. And there's a couple different ways to set the thresholds. I would say just very roughly, usually what I do is mouse over the image where the background is. And hopefully you remember from our last Explore ZEN that when you mouse over at the very bottom of ZEN, you see pixel values. Maybe Nicole, you could point that out with the mouse to people. It's going to be blank.
Yeah, just here at the bottom. So anytime your mouse is over your image, you'll see the pixel value change. So when I'm here in my background, it's obviously different from when I'm here in a really intense pixel. So
what do we do for thresholding here, Nicole, on the background? Do you remember what it was? Maybe a 1,000, something like that?
Yeah, I think we went with either 500 or 1,000 last time.
Okay. So if we set our threshold, this is one way to do this, and it's very dirty way to do it to start with. We'll discuss a couple different ways to go about this. So now we can see in our scatter plot, there's actually a number of quadrants. You see the numbers 1, 2, 3, 4, and you can see a vertical line and a horizontal line that are running parallel to the main axes. So these are the thresholds that we've set. The first threshold at 1,000, the second threshold, the vertical also at 1,000. And the analysis that we're doing for colocalization is quadrant 3, which is the largest quadrant. And that's where we have pixels that are above threshold for both of these channels. If we look in the data table, we have statistics for each one of these quadrants. So the first row is the statistics for the beta-catenin channel alone. So the pixels that are represented there are below threshold for ZO1. The next row, we have the ZO1. So the pixels there are above threshold for ZO1, but below for beta-catenin. And then the third row is the actual colocalization. And you'll see that we have correlation coefficients there. Pearson-Manders straight colocalization, which is an R squared. So these are different statistical methods of measuring the degree of colocalization or correlation between the two pixels. That will change dramatically if we move the background around. So we set the threshold manually. Nicole, let's just show a couple different ways we can set the threshold, like clicking within the scatter plot and dragging around. And while we're doing that, maybe if we turn on the coloc mask so that people can see the impact that it's having. So the purple channel. So this is a mask. You can see it's purple now, or what is that, a fuchsia, a pink? What are we looking at anyway? There's some staining now in the image that shows where the colocalization is being measured. And as we drop the threshold, you'll see that mask increase and increase. So if we can drop that to the bottom left, Nicole, just so it overdoes it at some point and it's really obvious to people.
There you go.
Yeah. So the pixels that we're evaluating and performing the colocalization statistics on are changing as we change that threshold. And I guess the take-home point that you should definitely get from this, other than you can quantify your colocalization, is that these coefficients are somewhat meaningless when they're taken on their own. So if I looked at just consider for example, I look at a single cell that has cytoplasmic GFP and cytoplasmic RFP. There's no associated protein. There's just the two fluorophores floating around, free floating. they're going to be coincident. They're in the cytoplasm, so the entire cytoplasm will show colocalization. There's no biological significance to that. There's no binding, but you'll get a very good correlation between the two. So the only way that this makes sense is if we have two different systems that we want to look at. System A, where we have maybe a wild type, and system B, where we have a mutant, and we want to determine the degree of colocalization, and then we can compare these correlation coefficients, and then we have some meaning associated with them. One is greater than the other. Did we cover everything, Nicole? I think that's about it, right?
Yeah, I think the other thing to maybe mention is if anyone was interested in how these coefficients are calculated, there's a place in the help menu where you can go and see these equations, right?
Oh, that's right. Yeah, so you can hit
like thesis purposes or anything like that, you know.
Yep. So as long as you're in colocalization, you can hit the F1 key, and that brings up the coloc view, and then you can investigate what's going on here, including the math behind these correlation coefficients. And thank you for bringing that up, Nicole, because in anticipation that this is a question, of course, as to what do these things mean and how do I use them, I've added a review publication to the handouts. So in addition to our cleaning and disinfecting your microscope handout, we've got evaluating colocalization that will do some description of what these statistics are. Oh, there was also the fact that we wanted to do a subset of the image. So right now we're analyzing the entire image that is above the threshold, but if we had a tissue a section where we had some portion of the tissue that was, let's say, cancer and some portion that was wild type, then we wouldn't want to analyze the whole thing simultaneously. So it is possible to manually isolate regions of the image. So if we take one of those draw tools to demonstrate that, Nicole, and yeah, we can draw around something that is of interest. And you'll notice as soon as that's selected that the table reflects not just the whole image, but also this subset. So we have on the table, the first 5 rows now is the entire image statistics. And then there's another 5 rows that's just this region of interest that we've highlighted. Okay. I think we're good.
Great.
We covered everything. Let's just take a look and make sure that we haven't missed a question here. If I can steal the mouse from you, Nicole.
Yep. Go for it.
No questions as of yet. I'm surprised, I have to say, if not almost shocked that we went through that whole colocalization without any questions, but fair enough. I guess that means we did a great job of the explanation. So let's shut this down and we'll go on to spectral unmixing. So again, I think that this came out of a question that we had last time, although I followed up with the person who asked the question, and I think maybe that there was a misunderstanding. But by the time we sorted it all out, we decided that we were going to do colocalization. I think that we're doing mito. What are we doing here? Colocalization we did. Spectral unmixing we want to do.
Are we doing the 5 color or the 7 color?
It's the BPAE crosstalk, but we're going to start with BPAE crosstalk. So fifth button, 6th button, something like that across the top there. Yeah.
There you go.
Okay. So spectral
I got a
unmixing.
little lag going on here.
Okay. Actually, I'm going to take advantage of that lag, and I'm going to bring up my little discussion point here, if you don't mind, Chantal. So the question really that this originated from is, I have background in my image, how do I get rid of it? And that's a pretty common question, so I thought, well, this is something that we should address. So let's talk about what is background before we go to spectral unmixing, because the two things aren't necessarily the same. So there's 3 main places that I would say we can get background from, and they're very different in terms of how we want to deal with them in the microscopy. So the first way that we have background is maybe bad sample prep. And I'm not criticizing the user here. It could be that the antibody is just not so good. But whatever's going on, the background in quotes that we see is in fact the fluorophore of interest. So if that's the case, it's absolutely impossible for us to extract the background mathematically, because it's the same as the fluorophore. We can't unmix these things somehow as background or signal, because it's all signal. We can subtract it, of course. We can erase it, or we can change the display so it doesn't show because it's lower intensity. But you have to use your judgment as to whether that's an appropriate way of eliminating something that's in fact the same signal that you're looking at on your object. Okay, we have tissue autofluorescence. So if we have a wash, often this is in the green channel. I think that brain that we saw that we did the shading correction of was a good example. That whole brain is green to some degree. That's certainly not the intention of the stain, but that tends to be 488 channel in coronal brain slices, tends to be pretty messy with background. If we have tissue autofluorescence, then we can do something with spectral unmixing. So we can subtract out that autofluorescence using the techniques that we're about to talk about with spectral unmixing. And then the third type of background, again, putting that in strong quotes, is that we have some of the signal from one fluorescence channel, let's call it channel A, that's bleeding through into channel B. And that's maybe not considered background so often. It might be, it depends, but it's just it's crosstalk, it's spectral crosstalk. So the point of this is point 1, sample prep. We really can't use spectral unmixing. 2 and 3 we can. In order to distinguish between 1 and 2, we really need controls. So we need to eliminate our secondary antibody and the fluorophore. And if we still have this fluorescence wash in the background, then it means that it's something that's a property of the tissue, or another chemical possibly that we're putting on, but at least it's not the actual antibody we're labeling with. Okay, sorry, that took longer than I had intended. But let's take a look at if we have crosstalk. So what does this look like? Actually, the nicest way to see if we have crosstalk initially, because it's not always obvious just by toggling the samples, is to look at the colocalization plot, believe it or not. That's why we did colocalization before we did this. So if we click on colocalization, in this case, I have artificially bled the DAPI through into the mitochondrial channel. And if you notice the y-axis, the vertical axis, DAPI, as the intensity goes up in the DAPI channel, the intensity of those pixels also increases in the Psi3 channel. So this is a really important point. If you have bleed through, the amount of bleed through that you have in every pixel is always going to be linearly proportionate to the amount of the fluorophore that is bleeding into that pixel, right? So if I have more DAPI, I'm going to have always more signal in the Psi3 that corresponds, because it's a percentage of the DAPI signal. So in this case, when I manipulated this image, I took 20% of the DAPI signal and I put it into the Psi3 channel. And so that we're seeing this. Now we can unmix that because these two things are spatially distinct. We can ask the software to look at what's happening here, and we have tell it we have two distinct fluorophores, and that's what we're about to do. So this is a really quick and dirty spectral unmixing, and we're going to get progressively more complicated spectral unmixing cases, but we thought let's use a really simple case where it's very clear. So yeah, let's start with the unmixing. And Chantal is going to ask the software to find two different components in this image that have distinct spectra. So underneath the unmixing tools at the bottom, there's a button that says Auto Find ACE. And ACE stands for Automatic Component Extraction. And we have an option of telling it how many components we expect. But we can't say more components than we have channels, because each one of these is an unknown. Think of it like an A plus B equals C equation. You have to have as many equations as you have unknowns. Same here, we have to have as many channels as on the input as we have output channels. So the software has found two different spectra. This is not much of a spectra because we only have 2 channels. So we're not really getting curves, we're getting lines. But it's identifying the nucleus as one, and it's identifying the mitochondria as another. So we can now do an unmixing of this. There you go, linear unmixing. And we've colored our DAPI green, which is not really important one way or the other. We could change the color of it. But if we look at the colocalization, now you'll see that we've corrected this axis. So we no longer have a situation where every single pixel that has DAPI in it is now also has some intensity in the Psi3 channel. Chantal, do you think we can put these up side by side just as a regular 2D, and we'll toggle off the DAPI channel and we can see how it's bled through in the one and not in the other?
Can I do this here?
Yeah.
And
Just drag the other one in and then toggle off the first channel. Maybe zoom into one of them and toggle it off, and you'll see the oh, we're flipped around on the channels. Okay. May I take it for a second?
yeah, please go ahead.
So we'll do that, and we'll do that. So the right-hand side is where we had crosstalk, and you can see that underneath of the nuclear stain, the DAPI, we have a kind of a ghost image of the DAPI, and then on the left-hand side, that ghost image is gone. We only have the mitochondrial stain. Sound good? Okay, so let's do something a little more complex, shall we? We're going to do a 7-color unmixing now. Terrific. So this is going to open a number of images. And just before we go and do this, I want to take an aside again, if I may, Chantal, take the mouse from you.
Yep.
And do a little, just a single slide. Don't worry, this isn't going to turn into a lecture. I just want to make sure we're all on the same page about what's happening here. So on the left-hand side of this slide, I have an image, and I'm looking at a single pixel in this image. And that's really a key point here. When we do the spectral unmixing, we're looking at 1 pixel at a time. When I look at this single pixel, and I have multiple fluorescence channels associated with this pixel, so I've taken a multiple fluorescence images, and I'm looking at the 1 pixel that correlates to itself through all of them, I get a spectral curve as a result of that. So here's my experimental spectral curve. That's what this looks like. And I know, because I've added GFP and YFP into this sample, that this curve consists of some amount of GFP and some amount of YFP. But I don't know how much. That's the problem. That's where the spectral unmixing comes in. So in order to do the spectral unmixing, I have to provide to the software a reference curve for GFP and a reference curve for YFP. And once I have those reference curves, now this just becomes this linear equation where I have some amount of GFP plus some amount of YFP gives me my experimental data. In this case, the relative weights are 2/3 GFP and 1/3 YFP. And so I would end up with 2 channels, the intensity of which was 2/3 of what my experimental was for GFP and 1/3 for YFP. Okay, was that clear,
And
Chantal?
do you want to do a quick check for questions? There's nothing in the chat box, but I don't see the questions box. So maybe...
Yes. No questions to be had. We're still good.
Cool. Okay.
Cruising along. So what Chantal's opened here is a large tiled image. This comes from an AxioScan whole slide scanner. Can we see the original data, Chantal, before we go into the references? It's the first image tab. Yeah. There we go. And if we split that out, you can see that we've got 7 dyes on it. So they're labeled at the bottom: DAPI, GFP, YFP, Psi3, 594, Psi5, Psi7. There we are. And of course, there's all sorts of overlap here. The GFP and the YFP are overlapping. The Psi3 is overlapping to some degree. And we want to really have the individual channels We don't want to have this overlap. So we're going to unmix this. We can click on the Unmix tab if we would, Chantal. But in order to do this unmixing, we can't do what we did before where we asked the software to find distinct spectra, because we have no basis to suggest that these fluorophores are geographically distinct. That is, there's no pixels on here that we know for certain are only one color. With the DAPI and the Psi3 on the last image where we had the mitochondria and the nuclear, we knew that these things were distinct from one another spatially, so we could look at one pixel as the DAPI and we could look at another pixel as the mitochondria. Here we can't do that. We need to have negative controls. So that's what the other 7 images are that we have open. Those are the negative controls. If we open up any one of those, we can take a look at a split view of it and see that intensity is largely in a single channel in most of them. Some of them, we have some crosstalk. So that's the GFP. This is the 540. So we're going to have some unwanted intensity in 2 channels there. So on any one of these, well, in fact, all of these, we want to go to the Unmixing tab. And we want to highlight a region. We've done this before, so we already have the marker on it. So we have a curve. We can just use any of the draw tools to highlight the fluorescence here. It'll give us a spectra that is only this fluorophore. And from there, we want to save this curve to the spectra database. So Chantal, at the bottom of that Unmixed Tools window, just about 2 inches below your mouse, the button says Save to Spectra Database. Now, we've already done this before, so when Chantal clicks on the Unmixing Spectra folder here, you're going to see we've saved the DAPI, we've saved the 520. These are all the references that we've just opened. So we've already gone through this because we didn't want to do it 7 times in a row while we were online. But you get the idea. We have a single spectra for each one of these control samples. So let me be very clear on that. These are different slides that have been stained with only one fluorophore. And then we're using that sample that's stained with only one fluorophore, we're imaging it, and we're extracting the spectra that results from it. Okay? Good. So let's go ahead and apply these reference spectra to the data, Chantal. So we go back to the data, the original image. Yep. And now we're going to go down to this list, and we're going to hit the plus button in the bottom left-hand corner. And we're going to click on, yeah, there you go. We're going to add each one of these reference curves. So we started with DAPI, we're going to add the 520. We're going to go through this for 7 different images. 7 different reference curves, I should say. As Chantal's doing this, I want to point out one other option that's sitting to the right of the window that she's working with here. There's a button that says Create Residuals Channel. And the Create Residuals Channel, to me, is a very important feature. And you want to take a look at that, because the problem with spectral unmixing is it always gives you a result. You're always going to get something. And it's pretty easy to get something that looks good. Okay, so why is that a problem? Well, you don't know how accurate you are if you don't look at the residuals channel. If it gives you an image that looks good and you're happy with it and you go and you publish it and you don't realize that it's actually entirely inaccurate, it just happens to correspond to what you wanted to see, that's not really a good situation. So let's do this unmixing. We've got our reference spectra loaded in. We can see the reference spectra here. We've got our data ready to unmix. And what the software is going to do is go through that image pixel by pixel and determine the relative weights of intensity of each one of these curves, reference curves, and how much fluorophore from that reference curve is in that particular pixel. And what we're going to end up with is an image that has 7 channels on the output, plus 1 for the residual channel. But it's not going to have any crosstalk, or it shouldn't have crosstalk to the degree that it had at the beginning. You can imagine running through all the pixels in this, especially on a tiled image, takes some math. So we're processing away here at about 60%, which should be done shortly. We're doing pretty well for time. We're 40 minutes in. We're going to do Connect after this and Intellisys. Both of those are going to take a little bit of time, so I think we're looking at probably wrapping up about 3:15. All right. So here's our unmixed image. Terrific. And we can either do a split view or just toggle back and forth between them. And we can see that we have something that looks very different than we had when we had the full 7 channels independently. Do you want to do a split view of that, Chantel, or?
So it is.
Sorry, I meant a multi-view. My bad.
Oh, I'm sorry.
I don't know. What do you think the best way is to demonstrate that to people? I think either a multi-view or just toggling back and forth between the split view, one way or the other.
So like this multi-view? Or do we want to do?
There you are.
I can't do a split here, right?
No, you can't split it within the multi-view.
Yeah. Okay.
All right. Okay. Very good. So it certainly is possible. I think we've seen data sets up to 10 fluorophores on a laser scanning confocal. This was done on a wide field system. It was done on a whole slide scanner, so it's certainly possible. I think the 10 fluorophore solution that I saw was done on a 980 that had a spectral detector. So it gives you a much faster and easier way to acquire spectral information. Not to say you couldn't do it with a camera-based system, but it's a little nicer workflow with the LSM. So it is possible to do. All of the immunologists out there who want to use their 7 fluorophores simultaneously now can go ahead, to my regret, I'm sure. Okay, we don't have any questions on spectral unmixing, so let's move on, shall we, to the Connect. Chantal, what do you think? Are we good?
Yeah, I think we're good. Let me just close all these up.
Nicole, is that you for Connect, or is that, oh, that's Chantal, sorry.
Yeah, it's me.
Okay. So we did those first 3 based on questions that we had last time. But the whole theme of the Explore ZEN idea is that we want to expose people who are attending this to parts of ZEN that we wouldn't usually be talking about because they're not commonly asked for, but maybe there's some real value to them. They're just, people don't know to ask, or they're just not related directly to instruments that we're usually working with. In this case, we're going to look at ZEN Connect, which is a relatively new module. So it's not that we don't show it to people. It's just something that hasn't been around for very long. Let's just, I guess we open this up before we start talking about what the concept of it is, Chantal? So why don't I?
Yeah, I think the only thing that we might have missed, Dan, is of all the modules that we have gone through to date, what was ZEN Light and what was ZEN Blue?
Oh, right. Good call. Okay, well, you open up the ZEN Connect and I'll just mention that. So the shading can be done with ZEN Light with no license. The colocalization is a module that even if you have ZenDesk requires an additional license, so there's a cost associated with it. The Spectral Unmixing is part of any licensed Zen, so if you have Desk or anything else, it's there. And Connect is actually free, so if you're really only going to use this with a microscope, which means that our demonstration we're about to give you is somewhat limited, but it's a really powerful capability with the microscope. So let me explain what you're looking at here. Connect comes out of this idea of wanting to make instruments work together more and more. So Zeiss has dozens of microscopes. In addition to optical microscopes, we have electron microscopes, we have X-ray microscopes, all of these generating image data. And quite often somebody wants to take image data from an electron microscope, for example, and use it with an optical microscope. It's the same sample. And traditionally you would just put the two figures side by side. But it can be really impactful if you can actually correlate these images so that they become different channels of the same image. That's what Connect comes out of. So here what you're seeing on the most zoomed out version of this is actually an iPhone image. So the iPhone image was taken with the slide sitting on somebody's lab notebook, and that was correlated with images that were acquired on a macroscope. So if we zoom in here, we've got our whole brain. Looks very much like what we did the shading correction on. It's not my data set though, so I'm pretty sure that's not. And then that whole brain coronal section that we have here, we have smaller images overlaid that are from a laser scanning confocal. And then the even smaller images, we've run that LSM in a super resolution mode, and those are overlaid as well. So you're zooming in and in, you always get the context of what's going on here. And if we want to bring up any of the original data, which is kind of a cool feature here, we can select any one of these images. Chantal, you're going to need the arrow feature for that. And double click or right click and say view data, and that will link us into, there you go, open images in ZEN. That brings up the actual image. And any modifications we make to this image here are going to be reflected in our Connect project. So even though it was born out of this idea of doing correlative microscopy, the way that I see this as being very valuable is that you have essentially a graphical image database. So if you're on a microscope right now and you have ZEN installed and you look to the right and you see that ZEN Connect window, you can create a project. You don't have to do anything. You just give it a name. It sits there in the background, and as you acquire images, if you reference your ZEN Connect project, you'll see the images in the context of where they've been acquired. This is assuming you have a motorized stage. And you'll have that ZEN Connect project, which will then allow you to link into all of the data, and you can gather preview images, and you can gather high resolution images. If you want to get crazy with it, you can take a snapshot of your lab book to see what your protocol is and correlate that with the slide that you captured, whatever it is that you'd like. Of course, if you want to use it for the intended purpose of correlating images off your optical microscope with some other system, that's great too. Chantal, I think we were going to show people, if they were using it for that, how they would do the initial alignment of the images, right?
Correct.
Did you want to do that?
Yeah, so I was going to use just this component here.
Okay. Can I describe what you're going to do as you do it?
Yeah, so I changed the opacity of the sample just so we could kind of see through it. And then I hit the align button here. And you can see that I can move this sample over what I'm looking at. And then you have to find, you have to find where these little guys are overlaid. And they are right here, I believe.
So
Do I have that right?
As Chantal's doing this, I'll just mention that if you're bringing an image from system A to system B, whatever that is, there's going to be an initial alignment procedure that you have to do. Once you've done that on one image, whatever translation, rotation, skew, etc., has to be applied to make sure that the coordinate systems are the same, gets automatically applied to all the other images. So this is a one-time alignment, one time per sample, so one time per slide alignment that you would do between the samples.
So I am trying to, this is why I haven't selected the translate, rotate, scale, and shear.
Great, okay.
And then I want to make sure that it's overlaid properly, and then I want to lock it into position with the right button on your mouse.
So you looked at that cell body, you said, okay, the cell body's overlapping, and then you right clicked to say, okay, there you go. Now I'm going to lock that in place so it's static, and now you can rotate, translate, shear, whatever the image, so that you get it perfectly aligned throughout the whole image.
Correct. I think I'm a little bit off, but I think you guys get the concept here.
Yeah.
And then once you've selected your spots, you hit finish alignment, and it should align your images one on top of the other.
Yeah. So if you were on a microscope and you were to go and acquire another image, it would build itself onto the original image, but you wouldn't have to do that alignment. It would apply that same alignment you've just done with all the subsequent images. Terrific. Good.
So, yeah, this is a pretty cool little tool here, and you can see that we have multiple images overlaid on this one sample, different modalities. So we have the phase contrast image, we have the confocal image. So yeah, I think this is pretty cool.
It is free, which always helps. So this capability that we've shown you now is free. I should say if you're using it on a scope, there is a paid version of it that you can then use Connect to navigate. So you could navigate via this preview image. And that's something that does cost more. But for the images to show up and build this kind of image database is something that there's no cost associated with. It's also possible to do animations both in 2D and 3D, so that once you have these multiple images, if you wanted to, you could do this zoom out all the way, zoom in, and navigate between them. And you can put that into a PowerPoint so that when you're presenting your data to people, they have 100% idea of what the context is of the data that you've pulled out of your. So you're putting up that image, and instead of saying, what is that coming from and where is it coming from, they can see absolutely that it's a coronal brain slice and it's coming from this region of the brain, and et cetera, et cetera. Okay. Not bad. Very for cool. Free. Good. We're going to do some image analysis then. And more specifically, we're going to do some machine learning image analysis. So if you were here last week, last Explore Zen, we stepped through the basic image analysis. Nicole's going to take the mouse now and take us through a comparison between classical or basic image analysis and machine learning image analysis. And we're going to do a walkthrough of how this is done, what buttons are pressed in Zen. But we're also going to talk about the advantage of the machine learning. I think this is a capability that's very new to people, I would expect. I would be surprised if there was more than one or two in our audience who have used machine learning and image analysis, if that. So I think to do this, we're going to start with the, mitochondria with noise image, right? And
before you move on, can you just double check for questions?
Yeah, good call. Let's take a look here. Okay, we got a general question that sometime we should talk about deconvolution. Very good. Oh, hold on. Sorry, folks. I had to scroll up here. We actually have 3 questions here. So one of the questions was for the spectral unmixing. Sorry about that. I swear I was looking here, but it was scrolling down for me. So instead of acquiring the reference spectra experimentally, could you use the emission spectra that's available for a fluorophore as a reference for unmixing? So something from the company or published numbers, for example. Short answer, no. 2 reasons for that. One is that it's not implemented to do that. So the software just doesn't have the ability, although I guess we could fake it somehow, but no. And 2, the more important answer is that the actual spectra that you get off of an experimental sample is not necessarily the same as what the published spectra is. And that's because of the local conditions, local chemical conditions of your sample. So it could be that it's smack on, but it could also be that you're off by significant amount. I'm going to say, nanometers, multiple nanometers on the peak or the shape of the curve because of the pH local redox environment, whatever that may be. I have done spectral unmixing on plants, which can be quite challenging, and the plant has a response to light. As you're imaging it, you can see the spectra of the chromophores in the plant changing their shape in response to the fact they're imaging them, which adds another layer of complexity to doing this spectral unmixing, of course. Okay. The next question was, the spectra are acquired using a 7-channel set with a multi-track acquisition. That's correct. It was a wide field system, so it was 7 sequential images from a camera. If instead we use the channel S splitting on a confocal, so this is the spectral array that I was talking about, is the unmixing result improved as the channel width in nanometers is decreased? Right. Very good question. Thank you. So the answer is maybe, a very definite maybe. I'll just address the idea of this spectral unmixing and the width of the channel. So we were looking at a camera-based system there. Presumably the width of each one of the filters was something like 20, 30, 40 nanometers as a bandpass filter. And it makes logical sense that if we have more granularity on the spectra, that is, we're sampling at 10 nanometer increments instead of 40 nanometer increments, that we would get better unmixing because we have a nicer, smoother curve describing our spectra. But there's even though it's intuitive, there's a misunderstanding about the use of the peak of the spectra. And often people think, oh, I'm unmixing GFP from YFP, and so I have to see something that is 10 nanometers apart or 20 nanometers apart. What's really more important is capturing the shape of the entire spectral curve. And so we actually have a sample here, an example, I should say, where we're unmixing FTSE and Cytox green. I believe if you look at the spec sheets for these, the bandwidth or the peak is 2 nanometers apart, and you can see it's green and green. But when you actually look at the shape of the curves, the shape of the curve looks different for the two. And so what you really want when you're doing spectral unmixing, and this is why doing plants, the one saving grace of doing plants, if you do chlorophyll, chlorophyll is a very broad shoulder on chlorophyll on the emission into the red. And so you can look at how much that shoulder you have compared to how much of the other fluorophore you have, and it's really the shoulder that's differentiating this. It's not the peak. I hope that makes sense. So the answer is it might give you a better response if you have better spectral resolution. It depends on your fluorophores. If you have something that is distinct in its shape of the curve, then you should be just fine with a 20 nanometer, even 40 nanometer bandpass. As you get smaller and smaller, here's the trade-off also. Your signal to noise is reduced. And signal to noise can be an issue with spectral when you get very, very small spectral windows, because you're of course eliminating most of the photons from that channel. You don't really have much data left if you really make it small. Okay, sorry I spoke for a lot there. And there was a third question, which was the deconvolution. So we're set. Thank you, Chantal. I'm going to undock this questions here, so hopefully I won't miss if anybody else asks. It's over top of my ZEN window, but I think we can manage anyway. Okay. So Nicole, we're going to do some image analysis.
Great.
Okay, good. So folks, we're going to start. Actually, Nicole, let's open the reference mitochondrial image. I think we have a mito without noise. Maybe we don't. BPAE reference. Try that if you would, just to the left of your mouse. Okay. So this is an image, very nice image, high contrast with the mitochondria, and pretty easy to quantify by regular image analysis. What we're going to do is compare and contrast the classic image analysis versus the machine learning. We're going to step through how the machine learning works, And then we're going to show how machine learning can really give us a big advantage over classic image analysis when we have a poor image. So we've got a second image that Nicole's opened up here, where we've taken that really nice image and we've added noise to it, so that we have a rather poor signal to noise. The contrast is down, and we're going to see how the classic image analysis and the machine learning perform compared to each other. All right. So let's do classic image analysis just to remind people how this works on the reference sample, if we could, Nicole. So yeah, we're going to use that as the basis, and we're going to select the configuration for Mito Classic. And hopefully most of our audience has been here for the classic image analysis, but let's just go into the setup and point out that the way that we're segmenting this image, or the way that we're identifying the mitochondria, is by an automated threshold. So we've said, hey, look, this is a mitochondrial channel we're going to look at in step 1, and we can skip down to step 3, which is the key step here, where we're identifying the fluorescence intensity of interest. There we go. And we've set this for an automated, there we go. We've set this for an automated thresholding, where we have an Otsu threshold being calculated. And because we have a really nice high contrast image, it's not having any problems identifying the mitochondria. So we can cancel out of this, Nicole. I think probably everybody's convinced that we can take a beautiful image and identify mitochondria. That's not a problem. Well, let's do now some machine learning. So the very first thing that we do with machine learning is that we have to train the software what is an object and what is not an object. So there's a preliminary step that we do before setting up the image analysis. And that's done under the IntelliSys trainable segmentation window here. And we say start training at the bottom of it. It's a very simple process, but we wanted to run through it so that you understood what was going on. We've already got an image loaded on the top right-hand corner. We can use more than one image in a training set, and it's not a bad idea to do so, actually. But all we need to do here to train the software is to say, hey, look, I've got some mitochondria, and I've got some stuff that's not mitochondria, which I'm going to call background. And in order to do the training, you take a paintbrush and you select, in this case, we've selected mitochondria, and we paint over the pixels that represent the mitochondria. And once we've done that for a number of situations, we select the background and we paint over the background. Now, there's some important points here to note. First of all, you want to have a rather steady hand and make sure you're accurate, because every pixel that you identify is going to be used as an instance. And if you misidentify the background as mitochondria, for example, you're going to have a hard time of things. But secondly, you don't want to identify all the same type of mitochondria or type of background, because the machine learning is, sensitive to the context of the pixels. So if I was to highlight, the way Nicole is, background that only had mitochondria to the left, and I did that throughout my entire image, and it was only with the mitochondria to the left of the background, then I would have difficulty when I tried to classify background that had mitochondria to the right of it, or to the bottom, or to the top. So you want to make sure, more or less, that you have a good representation of the different types of background, both close to the object and away from the object, and also multiple spatial representations relative to the object. I think we've got more than enough representation here. For some machine learning.
Having fun doodling.
Yep, yeah, absolutely. So if we were to do this, if we were to finalize this, we would say train and segment, but let's not do that, Nicole. Sorry to burst your bubble. I just want to keep us moving here and not have to wait for the training. So that could take just a couple minutes if we did the train and segment. But if we click cancel, we've already trained this previously. So you've got the walkthrough of how this works. We want to quit without saving. Yes. And now let's select the mitochondrial machine learning version and open up the reference image again, if you would, Nicole, because I don't want to spoil things here. Yeah, and let's set up the image analysis on this with the machine learning. So we say set up image analysis. Oh, something's gone wrong here. Okay, say okay. And we're just going to select side 3. There we go. Okay. So what we wanted to see was that this third step looks very different. We don't have a thresholding value anymore. We don't have this ability to do automated OTSU thresholding, for example. We have the ability to select our machine learning classification model and to select the channel within that model. And what's being done now is that classification is running and we're seeing the mitochondria identified. So I'm going to toggle off the fill. We can see we've captured the mitochondria all the way around. Terrific. Okay, Nicole, I won't touch the mouse, I promise. We're good.
You're fine.
So the what we're building to here is what is the power of this machine learning? Besides maybe being relatively easy to operate, why is it that we would want to use the machine learning over the classic? And if we do this analysis with an image that is very poor signal to noise, and that's what Nicole's going to do at the end here, and we compare the results between the two, I think that it's going to answer itself as to why you might have some power. So we can cancel out of this, Nicole. And then we're going to use the, yep, there we go, that guy. And let's run the whatever you want on it, classic or machine learning.
Okay.
So you run the analysis. And then we do have a duplicate, so we can run it in parallel, right?
Yep.
MITA with noise duplicate. So this is the exact same image. We just saved a copy of it. And we're going to run the machine learning on it so that we can sit these things side by side and show you how the two worked. I've suddenly got an unreasonable anxiety that somehow it's not going to come out the way that that it has the past half dozen times I've done it on this exact same image. Okay,
Perfect.
so shall we split the view of these two and then zoom in on each one of them and we'll see what the difference is? All right, so Nicole just made us 2 containers so that we can drag this. So the machine learning, remember, is on the right, and the classic image analysis is on the left. There we go, now it is.
It lags, sorry. All right.
And we want to resize these windows so that we're not seeing the table or the graph here because we're losing some desktop area.
Yep.
And then we can zoom in on each.
Okay.
All right, so from a high zoom, it looks okay. But as we zoom in here on the classic, zoom in even more on the classic if you would, Nicole, because I think that we're going to find, there we go. We just can't see it when it's zoomed out. So our ability to identify the mitochondria on the classic has really kind of fallen apart with the OTSU thresholding. Now, is it possible that we can do some other tweaking to this image in order to get it to look better? Absolutely. We could do more with the classic image analysis. We could do more with the machine learning. This is not to say that this is the be all and end all. If you're doing lots of image analysis and you saw what we did with this, you would know that we can do some smoothing and some pre-processing. But the power of this is that with the machine learning, you're already starting off halfway down that track. And we can improve this even more if we want to. So we have this really powerful ability to extract objects from relatively poor data. That's what the machine learning does for us that we don't get with the classic image analysis. Okay. Terrific. I'm double checking questions here. I think we're good. Oh, we were going to mention that we did a webinar on image analysis in general, and we'll post the link for that, or we'll send it out with a follow-up email. I think that was the intent, wasn't it, Nicole?
Yeah, yeah.
Okay, so we have a webinar that discusses image analysis in general, and I think the last third of it, it was my webinar, I think the last third of it talks about the theory of machine learning and image analysis. So if you're watching this going, well, that's really great, but I don't understand what's happening, that's an entirely reasonable reaction. We did not explain what's happening at all. There was no theory there. It was just, these are the buttons you press. And if you want to know the theory at a very high level, without too much detail, just the concept, then the link that we're going to send out is going to be great. We do have a question. After using IntelliSys, would you be able to extract the image for further auto counting of objects? Yeah, good question. So thank you. With IntelliSys, all we're doing is generating this mask, and it's not really extracting any data at all. So once we have the mask, then we're going to get whatever data it is through the image analysis wizard on the left here. So in this case, I believe we've asked for it to count the area of the mitochondria as a fraction of the image and the total intensity. And then we have, for the mitochondria, we have all the different objects here and the area of every single one of these objects. It's pretty meaningless because of the way we segmented it, but you can get all of the data out of this mask that you could get out of the classic mask as well, no problem. Okay, hope that answered the question. Good. I think we're on to my bit of the show, and we're almost at 3:15 when I said we were going to wrap up. I'm going to try and make that. I'm not going to make 3:15. Who are we kidding? But I'm going to try and come close to that. So not too much longer. Most of this is just a tease. I'm going to close these images down, and I want to talk about two different things. And one of them is very brief because I'm not sure that it's of much interest to a large part of the audience. And the other is very brief because we're going to touch on it next week, but I want to give you the option to see it yourself because you have access. So the first one is scripting in ZEN. There is the ability to do Python-based programming in ZEN. It is a paid module. You can go Tools, Modules Manager and take a look to see if you haven't licensed on your particular. I would suspect that you probably don't because it's not popular, but here we are. This is the macro editor. So we can do any Python-based coding in here that we want to. I'm going to show you how easy this is to automate some things because we have a recording feature. So if I wanted to add, for example, a scale bar and a circle of some sort to this image, there, I've done it. And that's the code generated as a result. So if I wanted to repeat that and repeat that, I can create a little button. I don't have to create a button, but it's kind of nice. Oh, let's save this as something first. Good. I can create a little button here that does that annotation automatically. That's what our buttons are that Nicole and Chantel have been clicking on. They're at the top here. I'll delete these annotations to show you how it works. Click on annotation, and it pops up the same scale bar and the circle on the image. That's a very, very basic example of what the scripting can do. If you are interested in this, the most current version of ZEN install DVD has a whole slew of macros included. So these are all the macros that come on the DVD. There's a whole slew of them here, lots and lots and lots. Also, if you go on the web and you just Google Zeiss ZEN OAD, which is Open Application Development, you'll see there's a GitHub site for this. So if you don't know what GitHub is, then it doesn't matter. Don't worry about it. If you do know, then you know exactly what that's valuable for. And there's also a little, maybe a nicer place to start is the Open Application Development news group. So this is a posting of all sorts of questions and answers on all sorts of topics of using Python scripting within ZEN for image analysis and for instrument control. All sorts of automation in here. And you'll see postings from me asking questions all the time because I'm a self-taught, very poor Python scripter. Okay, terrific. So that's the scripting capability, open application development. Should mention also if you're using MATLAB or any external applications, there's mechanisms to interface ZEN directly with those software packages. Reach out to your local account manager or imaging specialist. So the very final thing to discuss is that there is a online image analysis engine that Zeiss has been developing for some time. It's been public for some time, but it is not discussed in detail, certainly, I would say, by the local account managers and imaging specialists, because it's really been ramping up. And come the second week of June, we're going to have a real tight integration of this of this image processing, online image processing with ZEN. So this is Appear, is the website, appear.com. You can go to it and create a free account. Everything about Appear is free. There's no cost to it at all. And it is a repository of image analysis modules. So here are the public modules that you can scroll through that do all sorts of different things. I won't go through them at all. You're welcome to take a look at them. Each one of these does one very specific thing, everything from counting nuclei up to just adding noise to an image. There's some volumetric analysis stuff. When you select the module, you can download it to make it yours. So my workspace, I have some modules associated with my account now that are stuck, and you create workflows which you can then feed images into that spits out data to you. And the reason we're not doing this and we're not discussing this more is that every one of these that is currently web-based, where you run through the workflow and you set the parameters, here, my thresholding parameters on the left, for example, this is all in the Appear website. All of that is going to be integrated in ZEN, and you're going to be able to run Appear in ZEN natively. So still web-based, but ZEN will be interacting with the Appear website, and you won't have to pop out in order to see it. Okay, so hope you join us next time, and we will publicize this widely so that you can see our next Explore ZEN. Before we sign off, let me just check one last time, see if we have any further questions. We do not. So Nicole and Chantal, unless you have anything else to add on, I think we're going to wrap up our second Explore ZEN. Have we covered everything?
I think we're good. Thanks everybody for joining us.
Yeah, thanks everyone.
Thanks very much, folks.
In the future, I think it's great if you pass that along as well.
Terrific.
Stay well.
Talk to everybody soon.
See you soon.
Bye-bye.
Bye.